even 11 or 12 genes which are driven by just one promoter. You're getting my point. So you have just one promoter which is transcribing all the genes. So such a system is called an operon. Operon is a cluster of genes which are transcribed together. So that means there is one promoter which drives the expression of all the genes, but then they are translated independently. So they are transcriptionally fused because it's a promoter, one promoter. They do not have independent promoters, but they are not translationally fused. That means they are going to encode for independent proteins. Now the question is how, when the sequence is transcriptionally fused, that means one transcript, then how it is making independent proteins. So for that what you have to have is, for example, uh, yes, so for example, now the class knows what is a promoter, right? So you have the uh, minus 35, right? Then you have minus 10, then you have plus 1. So this is all promoter, right? Then you have, if you remember, I told you you have to have an RBS. You remember that? And then
Yes, ma'am. Ma'am, one question is that like promoter is a specific sequence or any sequence can be a promoter. It should be just a upstream to the TSS.
oh, but then I told you it should be specific in the sense that it has to have a conserved hexamer at minus 35. It should have a conserved hexamer at minus 10. You forgot.
No,
And
ma'am, that I remember, but the thing is that
so
like,
these
can it be like minus 20? Will it be just all the time at minus 10 and minus 35, or can it be at minus 20, minus 45, or something like that?
Yes, it can be at minus 20 will be too far, but sometimes it is there a little farther. Like there are, for example, in Mycobacterium, it has been reported that they do not have minus 35 at all, but then minus 10 is there. So there are a few exceptions that you have, for example, minus 10 की जगह पे you have a conserved minus 12 or minus 13. For example, in actinobacteria there is a TG motif at minus 13. So then those are exceptions. But in general, it should have a conserved minus 10, a conserved minus 35 in and around the same position. Okay.
Yes, ma'am.
So
Yes,
then,
ma'am. Yes.
when we talk about an operon, so in operon, again what you will have is an RBS, and again you will have an ATG, and again you have some sequence. True. And then you have TAG, and again you have an RBS. So what I'm trying to tell you is that in the sequence you will be able to find that there is one promoter, correct? So this is the promoter. This is the promoter. But then this is the ribosome binding site onto which the ribosome binds and translation happens. You remember that? Protein synthesis takes place. So
Yes, ma'am.
likewise, you have an initiation codon, then stop codon, but then you have again another ribosome binding site, and then initiation and then stop codon. So that is why even though there is only one transcriptional start site, but then you have different proteins made because there are three different ribosome binding sites onto which ribosomes can bind and carry out independent protein synthesis, because each of these proteins have an independent initiation and stop codon. Is that clear?
Ma'am, that is clear. But one question is that these genes are known as open reading frames. So if there is no, if multiple ribosomes are not binding, then in that case, if just one ribosome binds, then it will be just one amino acid sequence, and that will create just one protein, right?
Right, right. But because there are multiple ribosome binding sites on the transcript in prokaryotes, so this is a situation in prokaryotes. In eukaryotes, you do not have operonic configuration. Okay. So in
Also, in eukaryotes, the operons are not found.
no, very
Okay.
rarely. I mean, again, as I say, in biology, you will always find one or more exceptions. So it's very dangerous to make a generalized statement, but I would say most of the time it is common in prokaryotes. But
Okay, ma'am.
one or two exceptions you may find.
Achha, okay, ma'am. Okay.
So I hope this operonic configuration is clear to
Uh,
everyone.
yes, ma'am. Ma'am,
Okay,
I had a question.
okay, this is now
Ma'am, Dev.
Dev Priya. Yes, Dev.
Yeah, ma'am. So there was supposed to be an extra class, but I don't think that happened. So when will you like to schedule that class?
So what I did was I was taking like I was extending classes if you remember. So I was taking like half an hour extra. So I was thinking to complete the Operon, the gene regulation today, and in case not,
Ma'am, we have a quiz from 10.
yeah, let's see. So don't disturb me now. Let's finish it.
And ma'am, one more thing about the Viva. So can we have
You
it
need
on?
to wait, Devpriya. You are asking the questions in between.
Ma'am,
Let's
but we have a quiz at 10, so then I'll have to leave the class. We can't discuss at the end. Just wanted,
So we
just
can
one.
discuss at 9:50. Hold on, 9 to 9:50. Still we can continue.
Okay, ma'am.
Let me at least finish one operon.
Yes, ma'am.
Okay. So then, so in prokaryotes class, the operonic configuration is quite common, and there are various examples of operons. For example, lac operon is for lactose metabolism, trip operon is for tryptophan biosynthesis. Likewise, ara operon is for arabinose metabolism. Likewise, for example, even the degradation genes like desulfurization genes are present together as DSZ operon. Naphthalene degradation genes are present in the form of a naphthalene, a NAP operon, something like this. So let us start with lac operon. So when we look at the E. coli chromosome, so lac operon is present at this position, that means roughly at 15 minutes if you see as per the clock. So roughly at 15 minutes is the location of the lac operon. So what you see is that the lac genes are flanked by two other genes, SYN and MHP. And the lac operon actually consists of three genes, lac Z, lac Y and lac A. So lac Z encodes for beta galactosidase, lac Y encodes for lactose permease, and lac A encodes for galactoside acetyl transferase. There is a regulatory gene as well, which is called as LAC-I, and LAC-I encodes for a regulatory protein which is called as LAC repressor. What is important to note over here is that LAC-I or LAC repressor is transcribed by a separate promoter, so it is not part of the operon. On the other hand, LAC-Z, LAC-Y and LAC-A, these three genes are present together in the form of an operon because they are transcribed by one promoter, which is shown over here at the bottom. For lac repressor class, there is an independent promoter. So remember that lac repressor is not part of the lac operon. Okay, now let us look at the promoter sequence. So in the promoter sequence, what you see is that you see minus 10, you see that there is a minus 35, and you can now, because the class knows the minus 35 sequence, you can see that it is not very conserved. So TT, and then instead of G it has a, and then rest of it is fine, right? TT kaka. And in case of minus 10 it is TT and then TAAT. So few mismatches are there, but still it is conserved. Then over here is the transcription start site, which is, I told you that it has to be a purine. So it's an A or a G. So here it's an A. Then over here ATG is the initiation codon for LAC-Z, which is the one which codes for beta galactosidase. Upstream to this minus 35 region is the sequence, which is the binding site for another protein which is called as CRP. CRP is for cyclic AMP receptor protein. And what is cyclic AMP receptor protein? I'll be telling you in a second. What is shown in gray, which is shaded, is actually operator, which is shown as O1, and operator is the sequence onto which the lag repressor binds. So this operator region, in fact, in case of lag operon, there is one operator present internal to lag Z as well. So that means operator sequences onto which the regulatory proteins bind, they can be part of the promoter, they can be part of the open reading frame as well. So let us see the functions of the three enzymes. So beta galactosidase enzyme, it catabolizes lactose because it's a lag operon. So it is for lactose metabolism. So it catabolizes, that means catabolic means breakdown. So it breaks down lactose into glucose and galactose, but sometimes it converts lactose to allolactose as well. Lactose permease is for the uptake of lactose, and galactoside, its acetyl transferase, actually its function is still debated, but in the textbooks it says that it acetylates galactoside sugars other than lactose, but then its function is still in debate. This is the typical diagram which shows that how lactose can be acted upon by the enzyme beta galactosidase and can be converted into glucose plus galactose, or it can be converted into allolactose, which in turn can be converted further. Okay, so then how does this operonic configuration helps, and whether lactose is a preferred carbon source for E. coli. So glucose is a monomer, whereas lactose is a disaccharide. So glucose requires minimal processing and therefore it is a more desirable carbon source than lactose. So cell has devised a mechanism that if glucose is available in plenty, then why one should waste time in metabolizing lactose, right? So the question is, when glucose is there in large amounts, then why the cell should spend time in this metabolizing lactose. For that purpose, that means glucose is a preferred carbon source and over lactose, right? So that means if you have in the media both glucose and lactose, then glucose is used by the cell first, and when glucose is consumed, then lactose will be utilized. Class, are you people listening or no?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. Yes.
So over here, if you see, this will result in what is called as a biphasic growth. When you have both glucose and lactose in the media, what is shown in the right figure, then glucose is used first, and when all glucose is consumed, then lactose is utilized. So this result tells you that glucose is a preferred carbon source by the cells, and when glucose is utilized, then only lactose is used. So then the question is, that means when glucose is used, right, that means when glucose is in plenty, that means during that time the lag operon must be off, right, because lactose is not getting consumed. The cell has devised a mechanism that when glucose is there in plenty, then why to waste energy in breaking down lactose? So the operon is turned off when glucose is there in plenty. So then the question is how this operon is turned off and how this operon is turned on. How is this regulation achieved? So I hope from this point this idea is clear that why regulation is important. If this operon would not have been under regulation, then ideally glucose and lactose both would be utilized at the same time. But the cell has devised a mechanism that why to waste energy. So for this purpose, such an operon are called as an inducible operon. Inducible operon, that means only when lactose is there, then the operon is induced. So lactose or allolactose are the inducers for the operon. And this operon is actually under dual control. First, negative control by a repressor, and I'll be telling you what is this repressor, and positive control by the protein CRP, whose binding site I showed you was present upstream to -35. You remember that?
Yes, ma'am.
So let's see. So what happens is that, as I told you, lactose is an inducer of the operon, because only when lactose is present, then the operon is induced. So how this induction happens? So if you now see the figure, so LAC-C, LAC-Y, LAC-A are the genes, P is the promoter, and O is the operator, which I showed you, onto which the repressor protein or the regulatory proteins bind. Okay. Over here is the LAC-I is the one which encodes for the LAC repressor. So what happens is that LAC-I will make the LAC repressor, which is shown over here, and LAC repressor will actually bind to the operator. When LAC repressor binds to the operator, then RNA polymerase cannot bind to the promoter. I mean, it will bind to the promoter, but then this LAC repressor, which will actually serve as a roadblock. And because it will serve as a roadblock, no mRNA of LAC-CYA will be made, and therefore no protein will be made. So that means in the absence of lactose, the operon will be switched off, because the LAC repressor is bound to the operator, which actually serves as a roadblock for RNA polymerase to transcribe the operonic genes. Right?
Yes, ma'am.
On the other hand, in the presence of lactose, so in fact the inducer is allolactose, but also lactose, and also another non-metabolizable sugar is used, which is called as IPTG, isopropyl beta D thiogalactopyranoside. IPTG. So in the lab, when you will be doing the experiment, we will be working on this promoter, and but then we will be using, instead of lactose, we will be using this non-metabolizable substrate, which is IPTG, isopropyl beta D thiogalactopyranoside. So what happens is that when this lactose, when this lactose, which is shown by the black triangle at the bottom, so when lactose is present, lactose binds to the LAC repressor, which is shown as I. LAC-I will make the LAC repressor. So lactose, when present, it binds to the LAC repressor and actually changes the conformation of the repressor. As a result of this, the conformation is changed, and therefore in this modified conformation, it cannot bind to the operator. Once it cannot bind to the operator, then RNA polymerase can bind to the promoter, and since there is no roadblock, it will actually make the LAC-ZYA. Is it clear, class?
Yes, ma'am.
Everybody?
Yes, ma'am.
Rest of the students are there or left?
Yes, ma'am.
Okay, good. Now let's see. So this is actually called
Ma'am, can you go to the picture there?
yes.
Ma'am, here we can see that leg I is perhaps in anti-sense strand. That's why it is being transcribed in different direction.
Mhm.
So, ma'am, like in which cases, like it should be in anti-sense strand, like in regulatory system, does it happens many times in anti-sense strand?
No, so then so in case of E. coli, it is like this, but there are reports. So for example, there are many cases where you will find in the same direction. So that we will discuss. Let's see if the time permits. So you can have convergent, divergent. So these are like divergent promoters, because one is in that direction, the other one is in opposite direction, right? So this is called divergently transcribed genes, because one is transcribing LAC repressor, the other one is operon. So this is divergent, but convergent is also there.
Okay, ma'am. Okay.
So what happens is that when you have high glucose levels, then the lac operon is shut down. So the way the CRP system works is that, so glucose actually inhibits the enzyme adenylate cyclase, and adenylate cyclase is the enzyme which converts ATP to cyclic AMP. Cyclic AMP can actually bind to CRP, and therefore it is called as cyclic AMP receptor protein. It is also called as CAP. It's the same protein which is called as CAP, which is catabolite activator protein. So when glucose concentration in the cell is low, then cyclic AMP concentration is high, and when cyclic AMP concentration is high, then it can bind to CRP, and then it will make a functional CRP, and then it will activate the operon. So therefore CRP or CAP is actually called as it's a positive regulator of the lac operon, and it is also, so cyclic AMP receptor protein CRP or CAP is actually a global regulator. So what I want to tell you over here is that lac repressor, which was mentioned in the previous slide, lac I makes the lac repressor, and lac repressor is very specific to lac operon. On the other hand, CRP or CAP is a global regulator. It can regulate lac operon, and it can regulate many other catabolic or many other, in fact, anabolic operons as well. For example, operons for arginine biosynthesis are also regulated by CRP. So CRP is a global regulator. So the term global regulator and local regulators are used. So local regulators, that means which are specific to a particular gene set, and global regulators which can regulate a number of genes across the chromosome. So they are not location specific. Okay, CRP in case of lac operon is a positive regulator, but just to tell you that CRP can serve as negative regulator as well. So there are reports where it can serve as a negative regulator. So let's see in case of lac operon how it serves as a positive regulator. So CRP can bind to cyclic AMP, which is shown over here, but this actually happens only and only in conditions of low glucose, right? That means when glucose is not there or when it is in very less amounts. And I told you the reason, right? Because high glucose inhibits the enzyme adenylate cyclase, which converts ATP to cyclic AMP. Right, so that means when glucose is high, then cyclic AMP will be low, and when cyclic AMP will be low, it will not be available to bind to CRP, and when it cannot bind to CRP, it will not make a functional CRP. Class?
Yes, ma'am.
On the other hand, when you have low glucose, then you have high cyclic AMP, because adenylate cyclase is not inhibited. So you have high cyclic AMP which can bind to CRP and which can actually bind to the CRP binding site which is present upstream to the promoter. And what happens is that because when CRP binds, the way it helps in the way it helps as in increasing the number of transcripts is because it helps in the proper binding of the RNA polymerase. The binding of CRP actually promotes the binding of the RNA polymerase, and because of the high affinity of RNA polymerase to the promoter, now more transcripts are made, and therefore it serves as a positive regulator. So that means lac operon is under the control of a negative as well as positive regulator. So negative control is by lac repressor and positive is by CRP. So let us see this figure now. So there are 4 conditions possible, class. So in this case, what you see is that situation number 1, that when you have low glucose, that means glucose is less and lactose is available. So in this case, class, are you people following or no? You need to speak
Yes,
up.
ma'am.
Yes, ma'am. Yes,
Yeah.
ma'am. We are following.
Okay. So when you have low glucose class, so as we discussed that when you have low glucose, then CAP protein or cyclic AMP receptor protein will bind, because in low glucose you have high cyclic AMP, correct? And high cyclic AMP will make a functional CRP or CAP, and it will actually increase the affinity of RNA polymerase, and therefore LAC genes are strongly expressed. Also, when lactose is available, then if you remember, lactose will bind to the LAC repressor and it will change the conformation of the LAC repressor, and therefore there will be no roadblock. You remember that?
Yes, ma'am. Yes.
So therefore
Yes,
LAC
ma'am.
genes are strongly expressed. In case number 2, when you have high glucose, so when you have high glucose, that means you have low cyclic AMP, right?
Yes. Yes.
So when you have lactose is not there, that means the repressor protein will bind to the operator, and therefore it will serve as a roadblock and LAC genes will not be made.
Yes, ma'am.
In case number 3, when you have low glucose and lactose is not there, so even though you have low glucose, that means you can have high cyclic AMP, but because lactose is not there, so that means roadblock is there.
Yes, ma'am.
So because repressor protein can bind, so because of this roadblock, LAC genes will not be expressed, even though the glucose is low.
Yes, ma'am. But in this case, if lactose is unavailable, then why do we need like beta galactosidase and that thing?
We'll come to that. The fourth possibility is high glucose and lactose available. So in this case, when lactose is available, then you have very low or basal level of expression because the glucose is high, and when glucose is high, then cyclic AMP will be low, right?
Yes, ma'am.
So therefore you have only very low or basal level of expression. Okay, so now the question is, when lactose is not there, then why do we need beta galactosidase? Yes, because the cell has all of these enzymes, the genes are there only in cases when the preferred carbon source is not there. So most of the time, for example, glucose or simple carbon sugars are preferred, but then complex carbons like cellulose, like lactose, like galactose, so these are disaccharides or complex polysaccharides. So for utilization of these carbon sources, the cell has devised a machinery to switch the genes on only and only when required, otherwise they would remain off. It's another diagram which explains the same, and I have to tell you over here that the mathematics of recruitment, rejection, and a lot of, so people have worked a lot on lacoperon, and in fact Professor Narang in our department has contributed a lot, and even now his lab is working a lot on lacoperon. So there are still many things unanswered, so his lab has developed several models. So for example, the kind of repression that is achieved cannot be explained by inducer exclusion or by CRP based models. So he has proposed another model, and his lab is working on that. So if you people want to learn more about this, then his lab is the best choice. Okay, so then so hope lacoperon is clear to everyone.
Yes, ma'am. It is clear.
Yes, ma'am.
Okay, so the next, so lacoperon is an inducible operon. That means it is induced or it is turned on only and only when required. So people thought that such an operon, such a inducible operon is actually very useful, because for recombinant protein expression, if we use lacoperon like system, then it will be very desirable, because it will not cause any metabolic burden to the cell, right? So what people did was they used the LAC promoter Class, are you getting my point? So they use the lag promoter, they use the lag operator, they use the lag I, but instead of lag ZYA, what was done was they were replaced by a multiple cloning site. So that means the whole cassette containing the lag promoter and lag repressor was cloned in a plasmid, and lag ZYA were replaced by a multiple cloning site onto which you can clone any gene of interest. For example, you can clone insulin, you can clone erythropoietin, you can clone GFP, and what will happen is that instead of lactose, you will use, as I told you, the non-metabolizable carbon source, which is IPTG. So the moment you add IPTG, the operon will be turned on, otherwise it will remain switched off. So such a system class will not result in any metabolic burden, and therefore such a system can be used for regulatable expression of many recombinant proteins, which can be therapeutic proteins, which can be vaccines, which can be any other protein.
Yes, ma'am. Ma'am, one question was that, like, you described that Professor Narang is working on that. So, ma'am, for that, in that course, bioprocess also, this picture was there about lactose and glucose.
Mhm.
Okay, so, ma'am, about this is like like uh like people like Monod model we have learned.
Right,
So
but
they got Nobel Prize, right, just for developing that.
right. Don't say just for developing that. It
Yes, yes,
takes a while.
ma'am. Yes.
Takes a while to develop that, right?
Yes, ma'am.
So don't say just for developing that. For developing a model, it takes ages.
Yes, ma'am. That maybe I don't know.
But Monod's model so is being challenged now, and I think Professor Narang, I think if he has taught you, fine, but otherwise, I think there is another course, Dynamics of Microbial Systems or something, which also talks about these kind of systems approaches. Okay, so then the next operon is repressible operon. So it's 9:35, Devpriya. I will stop at 9:50 and then we will resume discussion. Don't worry. So the next
Yes, ma'am.
operon is a repressible operon. So repressible operon class is the one which is for amino acid biosynthesis. So, so in case of sugars like glucose and lactose, we have a system which is inducible because lactose is required. I mean, when lactose is there, then only you need to induce the operon. But for tryptophan, I mean for amino acids, so the system is such that when E. coli is growing in tryptophan containing media, then these amino acids can be taken up by the E. coli cell. On the other hand, when tryptophan is not there in the media, then the cell has developed a machinery to synthesize it. Okay, so this is now talking about synthesis. That means this is talking about anabolism, right? The earlier case was catabolism, that was breaking down lactose. This
Yes, ma'am.
this is an anabolic operon. Tryptophan operon is an anabolic operon because it is used for the synthesis of tryptophan. There are 5 genes which are responsible for the synthesis of tryptophan. So they are named as A, B, C, D, E. So the reason I am reading it in the reverse direction is because it's easy to remember. It's A, B, C, D and E. You need to write it in opposite direction. That's it. And then over here is a region which is called as trip leader peptide, and over here is the promoter and operator region. So most of the time in E. coli, cell has actually all the genes, all the operons for biosynthesis of amino acids like tryptophan, like phenylalanine, like for example asparagine. But in case, so these operons they remain off when you have tryptophan in the media. then the cell has ability to take up tryptophan, the cell has ability to take up arginine, the cell has ability to take up phenylalanine, right? But when you do not have these, then it can make its own. But then let's see how does it make its own. So the way it makes its own is the starting compound is corismate. Corismate is converted into anthranilate, and then after a series of reactions you see that L-tryptophan is formed. There are various enzymes involved which are tryptophan synthase, alpha subunit, beta subunit, anthranilate synthase, component 1, component 2. So likewise all of these, all the 5 trip A, B, C, D, E encode for different enzymes which can lead to biosynthesis of tryptophan from the starting compound corismate. What is important to see over here is that in case of tryptophan biosynthesis, the operon has 5 genes, point number 1. Point number 2, it has a leader sequence, and this leader sequence is actually translated into a peptide which is called as leader peptide. Now, what is a leader peptide? What is a leader mRNA? You will realize the importance of this in a second. And this is the promoter, the -10 and the -35. So these I have underlined just to show you that you can now see how conserved these are. So -35 you can see TT-GACA, and -10 you see that it has a few changes. So this is the typical diagram showing the trip operon control. So what happens is that if you see the yellow triangles over here, so yellow triangles show tryptophan. So tryptophan can bind to the repressor, which is trip repressor, and when tryptophan binds to the trip repressor, it makes an active repressor, and when it binds to this, then actually again the tryptophan is not synthesized because again it serves as a roadblock. On the other hand, when tryptophan is absent, then the repressor is not functional, it is inactive, and in this case the genes are on. So I have another figure for this, which is a better figure. So this shows clearly the operons are A, B, C, D, E. There is a promoter, there is an operator. So when tryptophan is present, the trip repressor binds to the operator, and then the RNA synthesis is blocked. So tryptophan can bind to the repressor. So tryptophan is actually called as a co-repressor. Class? And it can
Yes, ma'am. Yes.
and it can make a functional repressor, and when you have this binding, then RNA polymerase cannot make. On the other hand, when tryptophan is not there, then the repressor cannot bind, and therefore you have transcription. So this is one mechanism of control, that when tryptophan is there in plenty, then why to make the trip mRNA, right? Because when tryptophan is in plenty, then the cell can E. coli can take up tryptophan from the media. So in this case, why to turn on the genes for tryptophan biosynthesis? So the trip repressor binds to the tryptophan, and then it serves as a roadblock. Now let us see. Now let us see the other mechanism of control. So let me make this figure big. So over here are the structural genes, the trip A, B, C, D, E, and here is the regulatory region. The regulatory region actually has, if you see the -10, -35 promoter, the +1, and what is shown over yellow has a small region which is called as an operator region, onto which trip repressor plus trip co-repressor binds to make a functional repressor, which can bind to the operator, which is shown over here. And this region can also make a leader peptide. It can actually transcribe a leader mRNA, because the +1 is over here, and the +1 is actually the transcriptional start site. So what is important to see over here is that at the +1 transcription start site, the leader mRNA is made, and this leader mRNA has regions which are numbered as 1, 2, 3 and 4. The unique features of these regions are 1 and 2 can pair, 3 and 4 can pair, likewise 2 and 3 can also pair. So that means the sequence is like that 1 and 2 can pair to form a stem loop, that means a hairpin loop. 3 and 4 can also form a hairpin loop, and 2 and 3 can also form a hairpin loop.
Yes, ma'am.
Also in this sequence, in the region 1, there are two sequences which can actually code for tryptophan codon. So the codon is for tryptophan. Two codons for tryptophan are present. Okay. So over here in the sequence, again if you see, so in the region 1, these are the two codons, UGG and UGG are the tryptophan codons. And this is how the hairpin structures can be made: 1, 2, 2, 3, and 3 and 4. Now what happens is that this process is called as transcriptional attenuation. So what happens is that 1 and 2 can pair and 3 and 4 can pair. So class, when 3 and 4 pair, then 3 and 4 pairing can lead to a transcriptional termination. And the reason it can lead to a transcriptional termination is if you see the sequence. So what is there in the sequence? So what kind of transcriptional termination can you see over here? I have taught you transcriptional termination.
मैम RNA regulated.
नहीं सब भूल गया क्लास को. This is row independent. You remember that? Row independent.
Yes, ma'am. Yes.
हाँ. So this is row independent transcriptional termination, because in row independent transcriptional termination, remember I told you that you have a GC rich hairpin loop and then you have a poly A tail. याद है?
Yes, ma'am.
Yes,
So
ma'am.
then you have use, and then use can actually, so this RNA will actually get destabilized. So this DNA RNA hybrid will get destabilized and this will actually terminate the transcription.
Yes, ma'am. Yes.
So when 1 and 2 and 3 and 4, when these two form the hairpin loop, then actually the transcription will be terminated. On the other hand, class, on the other hand, when the pairing happens between 2 and 3, it will result in a hairpin structure like this, and this will actually not result in termination, because you see that this hairpin loop is not there, because 3 is not available for pairing with 4. 4 is separately there, and 3 has paired with region 3 has paired with region 2. So therefore this sequence is called as anti-terminator, and in such cases when anti-terminator hairpin loop is formed, that means when 2 and 3 will pair up, then you will not have transcriptional termination. On the other hand, when 3 and 4 will pair up, then you will have transcriptional termination. Is it clear?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Okay. So let us see what happens. So when you have high tryptophan class, so if you remember in the figure over here, if you see that there are in the region 1, there are two codons for tryptophan biosynthesis class. So what happens is that when you have high tryptophan, right? So when you have high tryptophan, then high tryptophan, that means you have more amount of charged transfer RNA which is charged with tryptophan class.
Yes, ma'am.
So that means you have tryptophan containing tRNA available in plenty because you have high tryptophan. So therefore
Yes,
this region 1 will
ma'am.
be transcribed rapidly, and therefore this region will be transcribed rapidly over here if you see, and therefore the transcriptional the over let me see over here, right? So you have this region will be transcribed rapidly, and therefore you will have the hairpin structure formed between 3 and 4.
Yes, ma'am.
And when 3 and 4 will be formed, then you will have the transcription will be terminated because 3 and 4 is a terminator.
Yes, ma'am. Yes.
On the other hand, class, when you have low tryptophan, then the UGG, the tryptophan codons are there. So when you have low tryptophan, then the tRNA, which is loaded with tryptophan, will be less in amounts. Class. So when you have the
Yes, ma'am.
tryptophan charged tRNA will be less in amounts. So what will happen is the ribosome will actually stall because it will wait for the trip loaded tRNA.
Yes, ma'am. Yes.
And when it is waiting for the trip loaded tRNA, during this time the hairpin between 2 and 3 will pair up because it is stalled at region 1 only. Because region 1 contains UGG, the trip codons.
Yes, ma'am.
So it will not move any further because it is still waiting for the trip loaded tRNA, trip charged tRNA because the tryptophan is low. Right?
Yes.
So in this case 2 and 3 will pair up, and 2 and 3, as we discussed here, that this is an anti-terminator. That means it will not result in termination. So therefore RNA polymerase will keep transcribing the mRNA, and therefore tryptophan biosynthesis will continue. That means when tryptophan is low, the cell will turn on, the cell will keep making the tryptophan mRNA, and therefore the tryptophan biosynthesis genes will be on, and therefore the tryptophan will be made by the cell itself.
Yes, ma'am. Yes.
Right?
Yes.
This is also a similar figure which is showing the same, but I thought this one is an easier one to follow. But
Yes.
otherwise in this figure also they have shown the same. But only thing is that in this figure you can see that you have a double stranded DNA, and then the pairing is happening between in the mRNA. But otherwise I think this is a better figure to follow. So hope this is clear to everyone.
Yes, ma'am.
Yes,
So
ma'am.
such a biosynthesis, so this, the two codons of tryptophan, these are called as attenuators, and such attenuators are found in many operons. For example, in phenylalanine you have, for example, 6 copies of phenylalanine. In histidine you have, for example, again 7 copies of histidine. In leucine you have 4 copies of leucine. Threonine, again you have multiple copies. So the cell has devised this attenuator mechanism for regulating the biosynthesis of the genes, regulating the biosynthesis of the amino acids, because by the presence of these codons you can have a control on the charged tRNA, right? So similar regulation exists for all other operons. Followed, class?
Yes, ma'am.
Yes,
Yes.
ma'am.
Everybody? Let
Yes,
me
ma'am.
check
Yes,
who
ma'am.
else is there. Dev Priya, are you there?
Yes, ma'am. I'm there.
Followed or no?
Yeah, ma'am.
Okay. Now, so it's 9:51. See, I stopped at exactly your time. Now you ask.
Yes. Ma'am, I was just asking about the Viva dates. Could it be on the 12th, 13th, and 14th? Everyone's fine with that.
Actually, 14th I do